So our goal with this was particularly to look at 1F slope and information theoretic measured, specifically the complexity entropy plane, to see are they complementary or redundant when it comes to looking at characterizing brain states. There's all kinds of brain states: being awake, asleep, all the different stages of sleep, being under different levels of sedation. And so we wanted to see, do these add information or are they redundant? In particular, we're interested in reproducing empirical feature data with the whole brain model, virtual twin, so that we can understand better what I like to call the virtuous cycle of experimental and computational neuroscience. So, okay, don't need this. It's like too many things in my hand. So we go from having a bunch of data, and from the data, we create models, and from the models, we run simulations to inform future experiments or clinical interventions. So in this case, we're using a whole brain model to understand the effect of varying degrees of propofol in the blood and what that does to brain dynamics and what effect it has on the 1F slope that I showed earlier, as well as the complexity entropy plane. So this will allow us to say, do interventions virtually before we can actually do them in a patient to get a better idea of what it will actually do. And then from there, you can do the experiment, collect more data, refine the models, run more simulations, and hopefully advance your treatment protocols or your experiments that you do in the future.
So yeah, first of all, what we did is look for a dataset that have the conditions that we are interested in to explore. And we find this paper that has an open dataset, is from 20 subjects and 91 channels of eyes closed resting states with different levels of propofol in blood. So here we can see like the typical alpha change in the alpha power when you have more propofol. You can see that the power of alpha, instead of being in the occipital lobe, goes to the frontal lobe. And yeah, so you have the baseline, then you have more propofol, then the moderate, that is like the most propofol in blood in these states, and then the recovery. So this data was like for 5 minutes of resting state with eyes closed. So yeah. Then for the methods that we use to characterize the empirical data and that we want to also compare to the virtual brain model that we are doing is with the power spectrum first, that follows a power law. We know that the electrical brain signals like the LFP, EEG, ECG, and even like magnetoencephalography follows this power law, or 1 over F to the B exponent, where B is called the spectral exponent or the 1 over F slope. And this has been proposed as a marker of arousal and cortical excitation inhibition balance. So here we can see the Gao proposed that when you have like awake states, you have like a flatter slope, that meaning more arousal or higher excitation. And when you have steeper slopes, you have lower arousal and higher inhibition. In this case, it's the same for a dataset of propofol. Here that you can see the same effect, that it has a steeper slope and the baseline has a flatter slope. This is also with rats data that happens the same effect. So yeah, that is what we want to replicate in this characterization of this dataset.
So to get more signatures about experimental data and simulation data, we will use the information theory. That basically is get the right signal, where is have measure to get time series, and we will associate to the time series on PDF. So after that, we can find the entropy and the complex. So the entropy and complex will be used to try to characterize different states of the different brain signal states. So for that, for find the PDF, we use the symbolic methodology of bend and prompt, and basically we will have some symbols, and these symbols will be the states of the time series, and basically the PDF will be how 8 symbols appear in these time series. So for that, we have 2 parameters. The first one is the event dimension, D, and it's basically how many points we are used to define the symbol. And then another one is empty time, it's basically which point we are see. For example, if tau is equals 1, we will analyze the neighbor points. But if tau is equal to 2, we are analyze the points with the step of 1. So after they find the PDF, you can calculate the entropy and the complexity. So the entropy basically is a mixture of information. So how many information we have of time series, and the complex is, for example, one mixture of symmetry, organization, and etc. So for example, here as an example, for the linear function, we have lower entropy and lower complexity. For the noise function, we have high entropy and lower complexity. But for the periodic function, it will in the mute of both. So for example, the general the bright signal is a sum of the periodic signals plus noise signals. So in this case, the brain signal will be in some place like there.
So to actually reproduce the empirical feature data to create virtual EEG data, we used the virtual brain, and we used the Zerlaut model using a data set that is based off of diffusion tensor imaging, and it has 96 different brain regions that are all modeled off of a mean field approximation to adaptive exponential integrate and fire neurons, where you can see the equations down here. So the problem essentially with doing this is that for adaptive exponential integrate and fire neurons, the adaptation term means that it is hard to do an analytical approximation, where when you're doing a mean field approximation, what you're doing is you're replacing the temporal dynamics, the precise things that are going on at the individual spike level, with an instantaneous firing rate. And this is very hard to do in adaptation in the ADX model, because the firing rate actually changes, getting slower and slower as it's getting more stimulation. It doesn't respond to every single input with the same exact output. It adapts. And so in order to get past this, the Zerlaut model actually took a bunch of these ADX integrate and fire neurons, well, more like a single one, and they ran it with a bunch of different parameters to get a statistical model of sort of what would be the firing rate in response to these incoming input currents from excitatory and inhibitory neurons. And using this, they're able to make a semi-analytical solution, which allows you to effectively get a mean field approximation model that allows you to capture the dependence of the firing rate of any particular neuron on the history of spikes that has come into it. And so you see here we have regular spiking neurons, fast spiking neurons corresponding to the excitatory and the inhibitory. Once we have this sort of mean field approximation for individual neurons, bring it into 10,000 excitatory and 10,000 inhibitory neurons, making that into a mean field approximation, because this is how you do it. And then finally, taking these and making these all individual regions inside a brain for a whole brain model. And so for the data set we used, there are 14 subcortical regions, and then 96 minus 82 cortex regions that we're modeling. So these are all modeled actually as kind of cortical columns, and we're modeling the subcortical regions as cortical columns, but it does the job. And so this gives you a signal, an external current signal, a simulated signal that's like a local field potential. And this is great, but that's more akin to an IEEG signal, an intracranial an electroencephalogram signal. And so we have to have a way to actually get EEG-looking signals that, actually are comparable to the thing that we see empirically when we do EEG. the best thing about EEG too is that you don't have to cut open someone's brain, and it's very cheap and accessible. And yeah, so it's great if you can get that from your model to make a virtual twin and therefore engage in the virtuous cycle I was talking about previously. The problem, mean field model averages out the kind of necessary data to produce empirically relevant data. And so what we did is added noise ourselves with pink noise. And the assumption being that pink noise captures the way that the local field potential permeates through the skull and captures the non-neuronal sources of noise that give you that nice spectral analysis or spectrum for spectral analysis in the future. Over here on the side, this is a forward projection model that actually details all of the regions in red, and for the black you see all of the channels. And so these are the precise locations of them inside the model and inside the empirical data that we use, where we had 91 channels, 96 regions in red. And so the 91 channels that you can see here are essentially listening to the entire crowd based on the inverse Euclidean distance weighted. So they're not getting the full signal from every single neuron, but a signal that's based on the particular distance that they are from the electrodes. And this gives you that signal that you'd like to have.
So yeah, moving into the results. For the empirical data, we did the 1/f analysis. This is the log-log spectrum of each condition for the 20 subjects. And then we feed it into the 20 and 40 hertz fit, because the data is only from 0.5 to 45 hertz. So we got these slopes. And then we use a toolbox that is called FOOF, and we did a linear fit, and we get this kind of linear fits that are the B exponent that we were seeing after the 1/f or to the B are these exponents of the linear fit that the FOOF algorithm does, and we are replicating the results of Gao and Colombo and a lot of other papers that find that you have flattened slopes for the wake or the baseline condition and more steeper slopes when you have like more propofol. So we get like the 1/f slope increases with the sedation level and being consistent with the GABAergic modulation of propofol.
So now for the information theory, we choose the amp dimension equals 6 and calculate the entropy and complexity for tau from 0 to 30. So here is for the average of the channels, and we find that the best tau for this data is equal 5. So look for the plan of entropy Vaxos complex. We can characterize the signal. So we see that for more propofol in the blood, we have more entropy and less complex. But in the baseline, that is before to take the propofol, we have more complex and less entropy. So now we can look for all channels, and we can define one distance from one state to the other. For example, here we have the distance, the Euclidean distance between the states and between the baseline and moderate. You can say that this distance is for almost channels. So here we can see the intensity of the distance. So and two, we can see that the distance is more high in these regions. So now look for the model. We try to apply the same conditions, and we find that for the average of the channels, we have a curve. It's a bit similar to the empirical data, but a little bit different. But we can as well reply the same behavior. So for example, here we have the parameters of the model. It just change these parameters. We can apply this behavior. So here is a compare of the model data and the empirical data.
So yeah, also we did like 1 minute of simulated data, and what we was with the noise parameters on all the default parameters of the Sasha paper. So it's like replicating almost the same model that Alain shows yesterday, and we only changed the tau value of the inhibitory synaptic decays and the adaptation parameter of the excitation of the population. So yeah, for these are the spectrum of the EEG data, and we had different fits, but in the 3 fits that we did, we can find like a similar like qualitatively fits. I mean, it's lower to higher slopes. So also the model data follows the empirical results. So only changing like biophysical parameters, this model is capable of doing like the signatures of true EEG. So yeah. And this also to respond like the question that we did about if 1 over f slope and complexity and entropy are saying the same things, we find with a Spearman correlation that is not significantly. And the thing that we did also a mixed effect models, but for the time I will just say that only the condition is what is moving the complexity and entropy and the slope. So they are saying different things about the brain state. So they are not redundant. It's like a good way to like characterize both of these metrics. So yeah, in summary, the model is capable of to replicate the same signatures of the empirical data, just moving like parameters that are biophysical in space. And here we can see like the same stuff there. And this like also a summary that the 1 over f slope systematically increases with propofol sedation, consistent with GABAergic inhibition, and our implementation of the ADEX whole brain model also captures these directions and for the entropy and complexity also. Yeah, and these metrics capture distinct and complementary features of brain states.
Limitations in future directions. So we had quite good fits. It's something like I think 5% off for our complexity entropy plane. I don't think we did a distance as yet to measure how far off our 1f slopes were from the actual empirical data calculated 1f slopes, but I think that we think that we can use simulation-based inference to actually achieve better fits, and that would be good to do for, say, if you're actually trying to make a virtual twin of someone. Well, you take their EEG, you measure the 1 over f slope, you measure the complexity entropy of the signal, and then you use simulation-based inference to actually fit the biophysical parameters to them, and then you can use that to make your tests. We also want to tease out what exactly the complementary information that the 1 over f slope is telling us. We have some sort of kind of idea about the complexity entropy plane, but with the 1 over f slope, there's a lot of contention in the literature over whether it's actually representing just excitatory and inhibitory balance, or if there's something more that it can tell us. And so in this case, it seems like maybe there's something more that it can tell us, and you can use that, both of them together, to characterize different brain states possibly more accurately than you can with just one or the other. We also want to understand why adding pink noise in the way that we did works so well, especially across conditions. We have the baseline, the recovery, the mild, the moderate, and it's the same noise for every single one. And adding that just worked perfectly. No other changes to the model besides tweaking these biophysical parameters of the adaptation and the tau I decay time. So, extending to other brain states would be really awesome, and also exploring topographic differences with our now EEG signal to see the different areas, maybe understand the functional connectivity dynamics, so alpha anterioralization, transient hypofrontality, and things of that nature. These are our references. Thank you!
Thank you. Have time for one very quick. Oh.
Okay, very quick. Nice, but and very quick question is: can you say again at what level you add pink noise?
We add pink noise to the excitatory firing rates that are coming outside of the model, each region. So.
Okay, so you don't put the noise in the equation; you just put the noise up once were solved.
Exactly. So it's like really part of the EEG and not part of the model.
Okay. Yes, that's what I was.
EEG level. It's really, we don't change the model dynamics at all, and it's just the way we're looking at what's coming out of the model.
It's just a quick remark is that exactly what you have done could be done also for psychedelics. Not because the model also exists for psychedelics, but also because there are data in open source of both EEG and fMRI of people under psychedelics, and I think you could just do the very similar analysis. And I don't think they have looked at the 1/a slope, by the way.
Yeah, it was to the output, only the pink noise. Yeah. Thank you.